Okay. Then the second part about the sperm is about physiology changes, capacitation. Okay, the sperm need to undergo capacitation before they can activate the ovum. Okay, the ovum. Okay. Wait, this one I will discuss later. Capacitation occur. Ejaculation. Okay. Okay. Between tubes, fallopian tube, the same. Okay. This slide here relate with these slides. Okay, this one. In terms of numbers of sperm ejaculated, okay, millions, hundreds and thousands of sperm ejaculated, okay, into the female reproductive tract, okay. The higher the number, the higher the percentage of successful fertilization or penetration. Why? Because sometimes only one sperm will only penetrate halfway, and then the sperm will dies out, and the next sperm will continue the second halfway. Okay, in that order, the path of penetration, or the path of penetrating the ovum, it's more, it's more easily and more readily for the successful penetration. Okay. Maybe when we look into the picture, then we can understand by this meaning. Okay, successful penetration of what depend on how many sperm surrounding the egg. Okay, so many sperm contributed to successful fertilization, but only one sperm can penetrate the ovum. Okay. Apa lagi ni? Ni dah. Okay, ni dah. Okay. So now we almost there. We almost do this part here. Okay. We have covered this. We have covered the tract condition. Okay, we almost here. And now we have to really understand the structure of the ovum. Okay. Okay. Nucleus, cytoplasm, Vitelline membrane, perivitelline space, polar body, zona pellucida, and corona radiata. Okay, corona radiata. Okay. So first of all, we have to be able to label it. Okay. In order to label, we have to recognize. Okay. Excuse me. So nucleus almost the most inner part. Okay. And cytoplasm is also inside the the ovum. Okay. Vitelline space is the outer layer. Cytoplasm, plasma membrane. Okay, so this is cytoplasm, this is plasma membrane. Okay, the vitelline space here right before zona pellucida. Okay, vitelline membrane, plasma membrane, perivitelline space. Okay. Maybe it will help you if you understand what is vitelline meaning, the definition of vitelline. Okay. Maybe someone can try to find the words vitelline, definition of vitelline, and post it in the chat box. Okay. And then after zona pellucida layer, okay, we have this layer here. The follicle cells, we call it corona radiata. Okay, cumulus osporus yang stuck tu kan. Okay. Okay, tadi saya itulah. Kalau you erase sekejap. Okay. Alright, polar body, we can observe here. Polar body is really it will be observable if the completion of second mitotic division occur. Okay. Okay, I put it in a bracket here, ZP1, 2, and 3 or 4. This is the term for the receptor for sperm binding. Okay, the sperm will bind to ZP1 or ZP1 or 2 or 3 or 4. In human, we have ZP3. Okay, in human, if I'm not mistaken, we call it ZP3. So this is the receptor for the sperm to bind. Okay, and then okay, ECM, extracellular matrix. So we have corona radiata. If you remember, it's a network of follicle cells that connected together. So extracellular matrix, and they contain hyaluronan or hyaluronic acid. Okay, hyaluronan or yeah. So this one will complement this structure here, the substance here, will complement with it's substances that can be degraded by the enzyme. That enzyme is in the sperm head. Okay. The sperm will contain enzyme that can digest the hyaluronan. That is the simple way. Okay, now we understand that we have corona radiata, and then we have zona pellucida, and then we have vitelline space. Okay, and we have the cell membrane and the cytoplasm itself. Okay, so really recheck the layers, corona radiata, zona pellucida. We have the glycoprotein, the receptors, and then we have the cell membrane. Inside the cell membrane, cortical granule. That's why I put. Okay, this is also a picture to help you understand. Vitelline layer, the plasma membrane. Okay, the protein receptor, the zona pellucida, exactly. Okay, uh, and we have the jelly coat, vitelline space, or yeah, right here we have the zona pellucida, or you can use other picture. Wait, I try to find it. Give me some. Okay, zona pellucida, but the outside or the outer layer of zona pellucida we have corona radiata. Okay, after zona pellucida we have ini tak ada plasma membrane, vitelline, peri-vitelline space. Okay, peri-vitelline space. Okay, so just remember the structure of these layers. Corona radiata, zona pellucida, peri-vitelline space, and we have the receptors, protein receptors. These protein receptors actually the ZP, okay, ZP1, 2, 3. Depending on the species, it will have different types of receptors. For human is ZP3, okay, 3 or 4. You have, have a read on reference book. And then we have the cell membrane, and we have the cortical granules, okay. It will really help you to understand the steps after this. Okay. So this is the phases of fertilization. You can use this slide, or you can use other slide, or the one that I show you just now, just to get the point here, and then you have to elaborate of each step. Okay, if you have to answer essays. Okay, the first one, the sperm squeezes through cells left over from the follicle. So these cells leftovers is actually corona. Here. Okay. So the cells over here is corona radiata cells. Okay. The ECM, extracellular matrix. So this sperm will penetrate, squeezes itself towards that corona radiata cells. Okay. So some sperms will dies, and then the next sperm behind itself or him, it will continue the penetration. Okay. So it doesn't mean only one. It doesn't mean that one sperm all along from cervix until penetration, that one sperm will become a successful fertilize or penetrating the egg, okay? Because they have their own lifespan, their own metabolic substances that enable them to become energetic sperm. But once that metabolic substances finish up, they cannot move, or they will become less motile and eventually dies out. Okay, after penetrating the corona radiata layers, they will meet the here the sperm acrosomal enzyme digests the egg's jelly coat. So now it almost enters the zona pellucida, the perivitelline space. Okay, just check in the reference book these jelly coats. Okay, so acrosomal enzyme, we call it acrosomal enzyme. Why this enzyme inside the acrosome? So refresh back our sperm's structure. Okay, we have the head, we have the mid piece, and we have the tail, right? And this the head, we have acrosomal cap, remember? So this acrosomal cap have enzyme that, so see, that's why these acrosomal caps at the most tip of this part of this jelly coat. This is also zona pellucida layers. Okay, it will expose the receptors. So these receptors actually the exact PZP. Okay, so exposing this protein receptor, eventually the sperm head also have the complement structure that can bind to this receptor. So it will bind. Again, I repeat, not necessarily the same sperm that digested the jelly coat that will bind to the receptor. Maybe the sperm number 2 already digested this part, but that sperm number 2 dies out. So the next sperm behind him, number 3, will have the chance to bind to the receptor. Okay. Just to really tell you the realistic story. Okay, sort of. Number 4, once the binding occur, that binding receptor binding will initiate other reaction. Okay, that will harden the zona pellucida. Harden. Okay. Okay. It at the same time, okay, bind, and then at the same time, once bind, it will fuse the membrane of this sperm. Cell membrane of sperm will fuse with the cell membrane of the egg. Okay, once fused, then the egg, the nucleus can enter. Okay, at the same time, okay, the zona pellucida will harden. Okay, harden. So this hardened zona pellucida is actually, we call it envelope, fertilization envelope. Nanti you akan come across with this word for, if I'm not mistaken, fertilization envelope. Okay, envelope. Okay, this hardening of this part over here, ni polyspermy dah lah. Okay, will prevent other sperm. Okay, remember when it's hardened, it's not hardened in this part, it will also harden at the whole ZP. Okay, so it will prevent any sperm to penetrate. Okay, because one sperm already fused with the cell membrane inside. Okay, next, number 5, the sperm nucleus enter the cytoplasm. At this time, at this phase, the egg, arrested magnetic division, metaphase 2, if I'm not mistaken, will continue will complete the magnetic division 2. Okay, will continue the phase. The anaphase 2 and telophase 2 and polar body. Okay. So fertilization envelope forms. This one, in order for this fertilization envelope, this is very fast steps. Okay, protein once the binding occur, and then it will fuse, and then at the same time, the zona pellucida starting to harden. Okay, so it's very. If we want to see step by step, this is the step 1, 2, 3, 4, 5. But in reality, once the binding occur and then the fusion, the fertilization envelope already, the harden of zona pellucida already occurring at the whole site, okay, of the oocyte. Okay, just now, this is the explanation step by step: penetration of corona radiata, penetration of zona pellucida and acrosomal reaction. The third one, fusion of the membranes. Okay, so this is the elaboration. Hyaluronidase enzyme contributed or come from the sperm, because the oocyte, the corona radiata has hyaluronin just now. Okay, so this enzyme will penetrate the ECM of corona radiata. Okay, this one comes from the sperm. Okay. Okay, flagella action also aids the corona radiata. So this is the factors that helps in the sperm penetration. Remember the flagella action. Okay, and then acrosomal reaction. Acrosomal reaction actually the release of the substances in the acrosomal cap. So it actually starts when it release the hyaluronidase. Okay. Um, if I want to say in terms of time span, okay, yeah, at the later part of penetration of corona radiata, the acrosomal reaction already starting to the acrosomal reaction is it's start. Okay, sorry. All right. Okay. What happened here? Zona is a glycoprotein shell. Zona pellucida layers. Okay. Will be penetrated by the substances in acrosomal. Okay. And then facilitate and maintain sperm binding induces the zona made up of 3 sulfate glycoproteins, ZP. Okay. So that it eventually receptors. Okay. Binding of that. Access to zona pellucida, but it will able to bind to the receptors. Okay, this is the substances or the enzyme contained in the acrosome. Penetration of zona pellucida around the oocyte. So we have esterase, acrosine, and neuraminidase. Okay, causes lysis of zona pellucida. So once exposed the zona pellucida, it will expose the plasma membrane. Okay, so this point here, permeability of zona pellucida changes when the head of the sperm comes into contact with the oocyte surface. Okay, once the sperm already able to make ways or penetrate the zona pellucida part here, and so the cell membrane of the oocyte already exposed. Okay, the sperm head coming into contact to the oocyte surface over here actually initiate the hardening of this zona pellucida that will prevent other sperm to penetrate. Okay, impermeable, okay, harden. Okay, so this is the real, if I've under microscope. Okay, acrosomal reaction, penetration of corona radiata, acrosomal reaction, and then you can see it start to penetrate zona pellucida or more inside. Okay, now we can see only the mid piece and the head and the tail. Yeah, at this stage here, starting here until here, it will start here. Okay, or the later or the end part of this stage, the zona pellucida is starting to harden. Okay. Next, okay, cortical reaction. Actually, after diffusion of the sperm plasma membrane with oocyte plasma membrane. Okay, this contact result, the contact of the sperm head with the oocyte surface. Okay, uh, initiate the lysosomal enzyme. Remember, we have cortical, so the cell membrane of oocyte, we have granules, right? Cortical granules. Okay, so sperm head appearing here, come into contact, this cortical granule will release the enzyme, the lysosomal enzyme. They release it. Okay. Okay, what happened? This enzyme alter the properties of zona pellucida. Again, to harden the zona pellucida part. Okay, prevent the penetration. So this is also polyspermy prevention. Okay, inactivate, plus it will inactivate the receptors. Okay, it will inactivate these gray color receptors here. It will make the zona pellucida and unable to be penetrated by other enzymes, by other sperm. Okay. Salah. Okay. Yes. Okay. Cortical prevention penetration, zonal reactions, the receptor. Okay. No further penetration. Also reduces the ability of the oocyte to, okay. And then Bertillon block. Okay. So yeah. So now we come into, so finish up this step. So polyspermy prevention. Just now when we go through these steps for the phases, we already cover a bit of the polyspermy prevention. Okay. The harden of the zona pellucida just now. Okay. Bertillon block. So that is also polyspermy prevents. It overlap. So the information over here that you know that zonal reaction here, we have to fit with this fast block and slow block. Okay, see cortical reaction. So prevention of polyspermy, there are two, fast block and slow block. Fast block is actually the membrane depolarization. Fusion of the sperm and egg plasma membrane causes depolarization of the egg membrane. So if we go through the steps, the picture steps just now. Okay. Okay, the fusion or the come into contact of the sperm head come into contact with the oocyte surface, and then eventually it fuse, right? So that will initiate membrane depolarization. Membrane depolarization is actually the disturbance of membrane potential. This one, we have to refresh our knowledge on excited cell or neuron. Excited cell. Okay, excited cells, example, muscle cells, oocyte. Okay, inside the oocyte more negative, outside the oocyte more positive. Okay, this membrane potential disturbed by the presence of the sperm head. Okay, the sperm now penetrate the layers and it come to contact in the it come into contact with the cell membrane of the oocyte. Okay, it fuse with this cell membrane. So this disturbance or abnormality, it will cause membrane depolarization, meaning the negativity inside the oocyte will become positive. Okay, membrane depolarization, if in muscle, it's about sodium chloride entering the muscle cells. Okay, for this membrane depolarization in oocyte, if I'm not mistaken, it's actually the calcium. Okay, so the negativity of this inside oocyte become more positive due to the calcium presence. Okay, any electrical charge changes. Okay. So that membrane depolarization is actually the first prevention of other sperm to penetrate the same egg. Okay. Why we call it fast? Because electrical charge, the changes, the negativity versus positivity. This is much more faster in terms of observation, or the changes observation that we can okay, we can observe under microscope, or the in terms of time span, if we make a slow motion of these fertilization steps, we can see that this fast block will occur first, that prevent other sperm penetrating the same egg. Okay, so fast block is the first one. It occurs due to membrane depolarization, the membrane potential disturbance. Okay, due to, if I'm not mistaken, due to calcium explosion. Okay, the slow block prevention of polyspermy is cortical reaction. So we go back to cortical reaction just now here. Okay, this cortical reaction releases many enzymes. Okay, the lysosomal enzyme, the cortical reaction, these cortical granules, or vesicles here, release its contents. So this enzyme will make a zona pellucida or Bartholin block. Okay, cortical reaction. It also release the calcium ion mediated cortical granule vesicle fusion with membrane. Calcium mediated cortical granules, meaning these cortical granules fused with the membrane is mediated by the calcium. So the membrane depolarization due to calcium, nak cakap apa? Entering or explosion. Please find out where this calcium comes from. But the membrane depolarization here occur due to calcium increase in concentration. Therefore, it will disturb the membrane potential. Okay, so this calcium ion inside this oocyte, the egg, will stimulate or will cause the cortical granules to release its content or fuse with the cell membrane. So it will hydrate the periventricular space and zona pellucida will harden. Okay, so go back to the structure here. It will harden this part. It will also periventricular space here. It will hydrate. Okay, so it will prevent other sperm. Okay. Okay, what else? It will break off the receptors. Okay, metabolic activation of A, calcium ion mediated. Okay, so this is fast and slow block polyspermy. Okay, this structure, the graph here just to show the membrane potential disturbance. The membrane potential is at negative 70 millivolts, but once membrane depolarization occur, it will become more positively charged. Okay, so you can see the sperm enter, suddenly it become depolarization. Okay, so it will prevent. In terms of this diagram here, okay, we can see here at constant membrane potential negative 70 millivolt. Suddenly the point of entry of the sperm, it will initiate depolarization. So every electrical current is very fast. Okay, it will distribute that membrane depolarization to the whole structure of the ovum. Okay. So again, slow block. Okay, I don't want to repeat back. I think that's all for today. Okay, the last part here, fusion of the oocyte and the sperm. This is about the fusion. Ya, ada calcium. Oh, calcium dah daripada sperm Okay, the last part is fusion of the oocyte and the sperm. About the fusion of the plasma membrane. The tails did not enter the sperm, the head, the nucleus enter the sperm. But the sperm plasma membrane remains behind. The sperm plasma membrane. It only fused and then it let the nucleus and the cytoplasm inside the head enters. Okay. Second method division. So once this fusion occur and so once the fusion occur, it will initiate the second metic division to complete. Okay. Okay, I think you understand this. And then form of pronuclei. So please take note this wording here, pronuclei. Is actually this one. Okay, the pronuclei maternal, paternal. So this pronuclei will fuse syngamy through fertilization. The unite of these two pronuclei. Okay, the true fertilization is actually fusion of the nucleus from the male and the female. Okay, the fuse. So that is the syngamy. That is the meaning of syngamy through fertilization. Okay. And that pronuclei is haploid. Both is haploid, one end and one end. Okay. The rest of it, I think you can read. Yeah. Okay. That's all for today. So it's only 4 minutes past 12. So far you have any question on fertilization? Is it all clear? Okay, tak ada orang jawab. Saya pun tak tahu.
Tak ada, madam. Clear je, madam.
Refresh ni. Tak best lah. Tak apa. Saya nak buat highlight sebenarnya. So remember, ni you tengoklah sendiri. Okay. So about transportation of the sperm, transportation and then physiology. So this transportation, it really helps to understand why this sperm need to be very motile, need to be packed with metabolic substances due to the female tract condition. Okay, a lot of obstacles at each part. And then you have to understand the opposite direction of the transportation between sperm and oocyte, and physiology changes of the sperm. Okay, ada dua. Pernah saya tunjukkan soalan kan? Soalan tu tanya. And then the phases. Okay. These phases is really helps to if we understand the structure. Lastly is the polyps prevention. Alright. Okay, that's all for today.